Right, so welcome to our online tutorial looking at the parts of microscope. Now, as this is a revision tutorial, you'll be just trying to remember back to times when in school you have utilized microscopes and the aspects of microscopes you need to know in terms of names, their function, and then in later tutorial as well, how you go about using them to prep slides and prep specimens. So in this one, we're going to look at parts of the microscope and also types of microscope. As you'll see on the screen, we've got a quite a basic drawing of a microscope that I'm going to go through and just make sure we understand the names of the different parts and what they do. So, if you remember when it comes to a microscope, there are two types of lenses. There is the eyepiece lens and the objective lenses. Now, um, obviously, some of this is kind of like quite self-explanatory, but let's go through it anyway. So the eyepiece lens, obviously, is the lens that you look down, and it typically has a magnifying power of times 10. Now, the objective lenses, if you remember from use in class, are the ones that obviously you can change magnification. The typical magnifications for objective lenses on these types of light microscopes are usually times 4, times 10, and times 40. Okay, now, main thing to remember is that the lenses, both of them, they make specimens appear larger. So, in other words, they are magnifying the image that you see. So they magnify the image that you see. Okay, so those are obviously the lenses. The area where you place your specimens and slides is what we refer to as the stage. So the stage is simply where you place your slide. Now, the other main component of standard light microscope that you use in class laboratories is this here that we often call the focusing wheel. Now, on some microscopes, there are two, a big one and a small one. And these are often referred to as the coarse and the fine. Coarse and fine focusing wheels. Now, their simple job in adjusting those, what you are doing to the image that is shown through the microscope is you are improving or increasing resolution. So in more simplistic terminology, that means you are able to get and see a clearer image. Makes the image clearer, and as a result of that, the person who's using the microscope is able to see more detail. Now, the other thing to remember is that when we're talking about these kind of microscopes, the microscopes you use in school, they're often referred to as light microscopes because the image is generated by using a light source. And that light source shines through the stage, through a hole in the stage, through the specimen, into the objective lens to create the image. Now, that means that the specimen that is used on the microscope has to be incredibly thin in order for the light to be able to move through the cells. If it's too thick, then the light can't move through the cells and you can't create an image that you can see. So, we've got a basic overview there of parts of a light microscope. So hopefully you'll be able to make your own version of this diagram for revision purposes. Now, as well as that, we also have to think about types of microscopes. So, there are two types of microscopes that you need to be familiar with. The light microscope, as we've mentioned, that uses light as a tool to be able to form an image that you can see. Then there's the electron microscope that uses electrons, which are negatively charged particles, sometimes here referred to as beta radiation, to form images of specimens as well. Now, there are some key differences between both of these. All right. Now, the first thing to bear in mind is that if you are using a light microscope, light microscopes have lower magnification and lower resolution, which means they can't create images as big and they can't create them in as much detail. So particularly you might see in light microscopes whole cells and seeing a level of detail up to whole cells. Whereas in electron microscopes, because they have got a higher magnification and a higher resolution, it means you can see images that are magnified to a greater degree in more detail. So it's got the capacity, for example, in cells to be able to see inside and the actions of subcellular organelles. So you're thinking about things, for example, like ribosomes or mitochondria. Whereas in a light microscope, you might be able to see that some of those organelles are there, whereas in an electron microscope, you'll be able to see actually what's going on inside of those organelles. Now, as well as that, other key things to bear in mind. So as we say, light microscopes, you've got the capacity to maybe see larger subcellular structures. So, electron microscopes, as we say, they use electrons to form an image. But because these electrons are fired at specimens, and obviously they're charged particles and can have the potential to damage specimens, you can only actually view dead specimens on electron microscopes. You can see them in a much greater level of detail, whereas on light microscopes, because you're only shining light onto specimens, you can actually look at living specimens underneath light microscopes. Now, the other thing to obviously bear in mind is that Size, electron microscopes are much bigger and than they are light microscopes. So with size, obviously comes cost. Light microscopes are a lot cheaper, whereas electron microscopes are much, much more expensive. So, key comparisons of the types of microscopes, and as well as that, we've also reviewed the parts of a microscope. So hopefully you've been able to make some useful revision notes from this tutorial, and thanks for watching.